1. Kingdoms, classification, nomenclature, and biodiversity. Kingdoms. Now there are 7. As you read the text that follows, you may come across words that are new to you. Most of them will be in the glossary. If even the definitions given there leave you scratching your head, I suggest you find and read a first-year university biology textbook before going much further. You may not know it, but you are about to become a member of an elite group. Although many people are aware that there are millions of different types, species, of living organisms on Earth today, although our own species is doing its best to drive many of them into extinction, surprisingly few people are aware that these organisms are now divided up among no fewer than 7 kingdoms. Before I can effectively develop the theme of this book, I must explain these major patterns and those of some of the almost 100 distinctive evolutionary pathways known as phyla. Botanists sometimes call them divisions, which make up those kingdoms. The really basic division among life forms is between the simpler prokaryotes and the more complex eukaryotes. The diagram below, after Patterson and Sogin 1992, shows the way in which we think the kingdoms evolved. It is based on molecular evidence, consisting of base sequences from ribosomal RNA, rRNA. The earliest forms of life, which appeared about 3,500 to 3,800 million years ago, were prokaryotes. We tend to define them by their relative morphological simplicity, although they have enormous biochemical complexity, and by the absence of many features found in more modern cells. Although their modern descendants have a single circular chromosome, this is not found inside a nucleus, and their cytoplasm contains no mitochondria or plastids, cytoplasmic organelles. These organisms make up the baseline kingdoms Archaebacteria, or Archaea, and Eubacteria. The prokaryotes had the world to themselves for 1,500 million years. They did, however, invent photosynthesis during that time. Not until about 2 billion years ago did life take the next giant step, the evolution of the eukaryotic cell. Many biologists now believe that this arose as a result of the mutually beneficial symbiotic union of several different types of prokaryotes within another host prokaryote. At least 2 types of modern organelles have been found to retain some of their original independent DNA. 1. Mitochondria, which specialize in the oxidation of 3-carbon organic acids, the Krebs cycle, providing an immediately available energy supply in the form of ATP, and 2. Plastids, which may contain photosynthetic pigments and enzymes. Chapter 1. Chloroplasts, or may store food. Some biologists also think that Flagella were once free-living prokaryotes, though they are very different from the motility organelles found in bacteria. Eukaryotic cells also have their DNA organized into a number of discrete chromosomes, which are found inside a nucleus which is surrounded by a membrane. Cell division in eukaryotes involves a complex process called mitosis. The nuclear membrane breaks down, a mitotic spindle of microtubules develops, and the chromosomes are duplicated. Then the daughter chromosomes separate and are pulled to opposite poles by the contracting spindle fibers. Each set of chromosomes then becomes enclosed by a new nuclear membrane, and the cell finally divides into 2. Prokaryotic cells have only a single, usually circular, chromosome and do not undergo mitosis. They usually divide by a much simpler process called binary fission. Mitosis, with its very accurate duplication and sharing of the genetic material, seems to have been a crucial invention. Only eukaryotic cells, with their precisely regulated genetic mechanisms, apparently had the potential to evolve into more complex, multicellular organisms in which cells are organized into different tissues and organs. All prokaryotes are still microbes. Now look at the kingdom diagram once more. The Archaebacteria, or Archaea, and the Eubacteria are prokaryotes. The eukaryotes encompass the other 5 kingdoms, and it is in these other kingdoms that the dazzling evolutionary explosion of new taxa has occurred. The diagram shows 5 eukaryote kingdoms: Protozoa, Chromsta, or Stramenophylla, Plantae, Animalia, and Mycota, or Fungi. New information on the fully sequenced genome of an Amoeboid, protozoan, pseudofungus, Dictyostelium discoideum, has now thrown some light on the timing of the origin of the kingdoms. The burst of eukaryotic evolution was made possible by, among other things, a modified form of mitosis called meiosis or reduction division. In many organisms, this produces special sex cells called gametes. Each of these sex cells has a single set of chromosomes. We say that the gametes are haploid. When two gametes from compatible parent organisms fuse, the resulting cell, the zygote, has 2 sets of chromosomes. We call this condition diploid. In plants and animals, zygotes develop into diploid, multicellular organisms, but in true fungi, the vegetative phase is always haploid, and therefore, meiosis must take place in the zygote. Whether meiosis happens in the zygote or at the other end of the life cycle, during the formation of gametes, it is responsible for the resortment of the genetic information built into the chromosomes by the process known as crossing over. New features are constantly being added to the pool of genetic material by the process of mutation, but sexual reproduction is the mechanism by which this pool is recombined. Each generation in most eukaryotic organisms, producing an endless supply of variation upon which the processes of natural selection can work. This is one of the key secrets of eukaryote diversity. The radiation of the 2 unicellular kingdoms, Protozoa and Chromsta, shows how the evolution of the eukaryotic cell expanded life's horizons. But the full potential of the new teamwork, I call it that, because several prokaryotes cooperate to make one functional eukaryotic cell, was not realized until cells, as well as cell components, began to cooperate. When organisms became multicellular, different cells could assume different, specialized functions. This division of labor eventually led to the kingdoms, classification, nomenclature, and biodiversity. Development of tissues and organs, and ultimately permitted the evolution of complex beings such as ourselves, beings with almost infinitely expanded capabilities, both wonderful and terrible. 3 new multicellular kingdoms arose, exemplifying 3 different ways of life. Multicellular organisms which could photosynthesize, make their own food from simple inorganic precursors, were eaten by other multicellular organisms that lacked this talent, and both were recycled after death by a third group. These groups we call producers, consumers, and decomposers, which roughly correspond to the plants, the animals, and the fungi. We recognize about 9 phyla of plants, about 32 phyla of animals, and 11 phyla of fungi, 2 Chromstan and currently 9 Eumycotan. The world being what it is, the picture is not as simple as we might like. Some of the divergent paths of evolution have come together again, almost as they did at the birth of the eukaryotes, and many organisms that seem unitary are, in fact, partnerships or even consortia. Lichens, for example, always incorporate both an alga, eukaryotic or prokaryotic, and a fungus. Other examples are reef-building corals and some medusae that coexist with endosymbiotic dinoflagellates, many sponges and some mollusks, and flatworms that coexist with endosymbiotic green algae, plants that often coexist with systemic endophytic fungi which may function in repelling herbivores, and almost all plants that also have mycorrhizal fungi intimately associated with their roots. How can fungi fit into 2 kingdoms? The answer lies in the way we define the term fungus, plural, fungi. Fungi are eukaryotic, their cells contain nuclei, heterotrophic, they can't make their own food, osmotrophic, they absorb, don't ingest, food, develop a rather diffuse, branched, tubular body, radiating hyphae making up nylia or colonies, and reproduce by means of spores. This describes not a single phylogenetic line but, rather, a way of life shared by 2 kingdoms of different evolutionary backgrounds. We recognize Chromstan fungi, or pseudofungi, as well as Eumycotan, or true, Fungi. If you find this strange, consider the algae for a moment. They include representatives of 3 kingdoms: eubacterial prokaryotes, the blue-green cyanobacteria, chromestans, brown algae, diatoms, etc., and plantae, green algae. Both algae and fungi are in fact defined functionally or ecologically, rather than phylogenetically. Recent additions to the true fungi. Whitaker, 1969, led to a general acceptance that true fungi constitute a separate kingdom of organisms, the 5th, but not the last, to be proposed. Yet the idea of a more or less universal hyphal module and microscopic spores as characterizing fungi led to their being mixed together with outgroups, such as the hypha-producing Oomycetes, which are primarily related to brown algae, kingdom Chromesta, the Hyphochitriomycetes. And, for no obvious reason that I can see, the Mixostellata, Mycetozoa, plasmodial slime molds, Plasmodiophorida, and Labyrinthulata, all 3 placed in kingdom Protozoa. These will all be briefly discussed later. Once these outgroups had been expelled, the true fungi were considered to embrace 5 phyla: Chitradiomycota, Zygomycota, Glomeromycota, Ascomycota, and Chapter 1. Basidiomycota. All of these are heterotrophic, lack plastids, and have chitinous cell walls. But those characteristics alone are no longer adequate to define the fungi, or, since some fungi, as you will see, have lost major characteristics, this definition may be too demanding. Blastocladio Mycota, a 6th fungal phylum. This group was considered to be an order within the Chitradiomycetes until 2006, when it was promoted to phylum rank based on molecular evidence. Microsporidae, a 7th fungal phylum. Molecular evidence has now added to the fungi a 7th, unlikely group of phylum rank, the Microsporidae, example Nosma, obligate intracellular parasites of animals, lacking walls in the somatic condition and long considered to be protozoa. There are 1,400 species of Microsporidae in 144 genera and probably many more. Why is the naked, mitochondria lacking, peroxisome lacking Nosma a fungus? Because it has chitin-walled spores and appropriate DNA sequences. During the course of evolution, the Microsporidae have lost many fungal characteristics, but their DNA gives them away. Neocauli-Mastigomycota, an 8th fungal phylum. The Neocauli-Mastigomycota, recently recognized and similar to aberrant Chitradiomycetes. Living anaerobically in the rumen of herbivores, and with multiflagellate spores, have also lost mitochondria and peroxisomes but are nevertheless true fungi. Cryptomycota, a 9th fungal phylum. These so-called hidden fungi turned up in a pond in England as unknown DNA. Once it was better characterized and visualized, this group was found to have cells 3 to 5 microns in diameter and to lack the chitinous cell walls so common in fungi. This meant that these organisms could feed by engulfing food particles, which is most unfungus-like, or live inside host cells. But then, rules in biology are often broken and we have to think again. DNA sequences typical of this new group have now been found in lakes in France, farms in the United States, and deep ocean sediment, and they may turn out to be very numerous, although they have not yet been grown in culture. It is also still possible that they are not really fungi. It is important to realize that the Mycota are not homologues of the true fungi, but highly convergent analogues. They have cellulosic walls and are somatically diploid, having arisen from a very different group of organisms, which includes the diatoms and the brown algae, kingdom Chromesta. It is now thought that fungus-like organisms arose at least 6 times walled somatic cells, no plastids, heterotrophic, but that only one of these became the true fungi, or Eumycota. Thus, the definition of a fungus is, more complex than we might wish. It involves a mixture of ultrastructural and molecular characteristics. Cilia, flagella, of true fungi, when they are present, as in the Chitradiomycota and Neocauli-Mastigomycota, never have flimers, tripartite hairs, like those found on Kingdoms, Classification, Nomenclature, and Biodiversity. Tinsel flagella, as in the Mycota. The cristae of true fungal mitochondria are flattened, like those in animals, while those of Chromsta are tubular, like those in protozoa. Fungi synthesize lysine by the alpha-amino adipic acid pathway, while Chromsta and plants employ the diaminopamelic acid pathway. A minimal definition of fungi might now be as follows. Absorptive heterotrophs, lacking plastids and therefore not photosynthetic, and not phagotrophic, that usually produce spores with chitinous walls and often have multinucleate, walled hyphae. I will close this brief outline with a caution. We tend to accept molecular evidence without raising too many serious questions, yet the outline of fungal phylogeny is still rather vague, because rRNA, the only molecule extensively used in constructing fungal phylogenies, evolves at such different rates in different groups of fungi that the phylogenies suffer from systematic biases as well as random noise. At this point, it becomes clear that this book does not, as its title implies, deal exclusively with the fifth kingdom, Eumycota, but also discusses some elements of kingdom Chromsta, and even the non-fungal slime molds. But these are all relatively minor players in the biosphere when compared with the huge numbers and biomass of the true fungi. Biological classification. The first part of this book deals with the classification of the fungi. You could certainly ignore chapters 2 to 7 and move quickly to the more accessible and, to many people, more interesting chapters later in the book which describe the many ways in which fungi impact human existence. However, I don't think I am overstating the case if I say that unless you understand something about how the main groups of fungi differ in morphology and behavior, you will not be able to make much sense of the more relevant sections of the book. If you can develop a sort of cognitive map of the main groups, recognize them on sight, and understand the unique abilities of each, you will find the study of fungi mycology infinitely more rewarding. A new phylogenetic classification of true fungi 2007. This new system has just been completed. It is based on all available information, but especially on molecular data. It is contained in a study that appeared in the journal Mycology titled A Higher Level Phylogenetic Classification of the Fungi by no fewer than 66 authors, Hibet et al. 2007. Note that it does not deal with the mycetes, nor with the various types of slime molds, none of which are true fungi. I am adding it here as a preliminary to reorganizing the first several chapters of this book. Please consult these tables so that you can place the taxa discussed later in their correct places. This first table deals with the 6 older and simpler groups of true fungi, including the Chitradiomycota, Blastocladio Mycota, Neocalimystigomycota, Zygomycota, Glomeromycota, and Microsporidia. Chapter 1. The second table covers the Basidiomycota, including rust and smut fungi. The third table covers the Ascomycota, including almost all lichens. E. Kixolales demargaritales harpalales acelariales zoopagales. Entomopterales. Blastocladiales. Mucora es endogonales mortierales. Neocalimystigales. Monoblepherus. Chitradiales spizellamistales rhizo H. dials. Archaeospirales diversispirales glomerales paraglomerales. Microsporidia. Kixellamycotina. Zoopagomycotina. Entomopteromycotina. Blastocladiomycota Blastocladiomycetes. Mucoromycotina. Neocalimystigomycota Neocalimystigomycetes. Monoblephyridomycetes. Chitradiomycota Chitradiomycetes. Glomeromycota Glomeromycetes. D. Eukaryote. Traditional. Zygomycota. Chitradiomycota. Table 1.1. Kingdoms, Classification, Nomenclature, and Biodiversity. AWIIEM1 AES, 1 AWIIEM1 Amis Est. Enterizales, Antarrhizomycetes. Classiculales, Classiculomycetes. Cryptomycoclicales, Cryptomycoclicomycetes. Myxiales, Myxomycetes. Atractyolales, Atractyolomycetes. Spiculogloeals. Agaricostilbomycetes. Agaricostilbals. Cystobasidiales 1 Erythrobasidiales Cystobasidiomycetes J. L _ Neohydiales. Puccinomycotina. Helicobasidiales. Platygloeals. Septobasidiales. Puccinomycetes. Pucciniales. _Picnocybulales. Heterogastridiales. Leucosporidiales. Microbotriomycetes. Microbotriales. Sporidiobales. // rec urocystales. Ustilaginomycetes. Ustilaginales. Malisozyales. Doasonciales. Ustilaginomycotina. Antilometal. Exobasidiales. Exobasidiomycetes. George Fiscariales. _microstromatal. Tiltials. // Cystophilobasidiales, 1L _Philobasidiales Tremolomycetes. / Tremelles J. Dacromystales, Dacromycetes. / Auriculariales. Sebacinales Agaricomycetes Scantharellales Trachysporales. Geestrals. Gomphals. Phallomycetidae. Hysterangiales. Agaricomycotina. Phylals. / Anagaricales hymenocetales. Corticials. Gloeophiles - Polyporales. Thellophorales. -- Russules. Elathalales Agaricomycetidae J. Bulltails. Table 1.2. Chapter 1. Neolectales, Neolectomycetes. Pneumocystidales, Pneumocystidomycetes. Taphronomycotina. Schizosaccharomystales, Schizosaccharomycetes taphrinales, Taphronomycetes. Saccharomycetes, Saccharomycetes Saccharomcotina. Orbilials, Orbiliomycetes 1 Pezizales, Pezizomycetes Lamiales Mediolariales Triblidiales Capnidiales Duthidiales Myriangles Duthidiomycetidae Pleosporales, Pleosporomycetidae R Botrios Phaereales Hysteriales Patellariales Jahulales Arthoniales, Arthoniomycetes Ketotheriales Pyranulales Ketotherlomycetidae Verrucariales Pezizomycotina Duthidiomycetes 1 Mycocaliciales, Mycocaliciomycetidae Uroshiomycetes Urocials Anigenales. Uroshiomycetidae Carinaliales Labulbeniales Pixidiophorales Labulbeniomycetes Lecanales, Lechinomycetes Acarosporales, Acarosporomycetidae Candelariales Umbilicariales Lecanorales 1 Peltigerales Lecanoromycetidae Teleshistales Agiriales Biomycetales Ostropales Pertusariales Sidereales Erysiphales Helatiales Retismetal Thelebales Calispheriales Lulworthiales Meliolales Philacarales Trichosphaereales Ostropomycetidae Xylariales. Xylariomycetidae Caranophorales + 1 Hypocreales Microascales Melanos Orales Boliniales Ketospheriales Coniacetales Diaportales Ophiostomatales Sordareales Hippocreomycetidae Sordariomycetidae Lecanoromycetes Leatiomycetes Sordariomycetes. Table 1.3. Kingdoms, Classification, Nomenclature, and Biodiversity. Biological nomenclature. Every species of living organism is a collection of individuals which are very similar, genetically, if not always in appearance, and each species has a unique name made up of two words, which may actually be from the ancient Latin lexicon but are far more often new, pseudo-Latin words coined for the occasion. This two-epithet name is the binomial. You and I belong to the species Homo sapiens. The supermarket mushroom belongs to the species Agaricus brunscens. In each case, the first of these two Latin words is the generic name or epithet. This places the organism in a genus, a collection of similar and/or related species. The second Latin word is the epithet applied to one particular species of the genus. But notice that the name of the species always consists of both epithets together. This is because only the two-word combination is actually unique to that species. The generic epithet is shared by all other species in that genus. The same species epithet may also be applied to species in other genera. For example, many Canadian spring flowers, although belonging to different genera, have the same species epithet, canadensis, as does the national animal of Canada, the beaver. Therefore, remember that only the two epithets together, the binomial, properly specify a species. Homo sapiens is the only extant species in the genus Homo, but most genera contain more than one species, and some, for example, the mushroom genus Cortinarius, are made up of hundreds of species. For purposes of classification, which is actually a method of information storage and retrieval, related genera are grouped into families, families are grouped into orders, orders into classes, classes into phyla, and phyla into kingdoms. Here is a sample of how an organism is classified in this hierarchical, boxes within boxes, system. Note that the binomial is in italics, as it is in most scientific publications, while the names of higher-ranking taxa, families, orders, etc., are not in italics. If you refer to a binomial in writing, it should be underlined to show that it would be printed in italics. Kingdom, Mycota or fungi phylum, Basidiomycota subphylum, Basidiomycota class, Agaricomycetes order, Agaricales family, Agaricaceae genus, Agaricus. Species, Agaricus brunscens, the edible, supermarket, mushroom. Why do we use this binomial nomenclature, which is so unfamiliar to the man or woman in the street? Why not use common names wherever they exist? For three good reasons. 1. The common names of many organisms differ from country to country, and even from district to district. 2. The same common name is sometimes applied to different organisms, for example, the British, North American, and Australian robins. 3. Common names can be downright misleading. Irish moss is a red alga, Spanish moss is a flowering plant, club moss is a fern ally, and reindeer moss is a lichen. Chapter 1. Let's face it, common names are too unreliable and confusing to be of any use to scientists, who rely heavily on international communication and cooperation. Please take the time to learn the proper scientific names of the more important organisms you encounter in these pages. If you ever want to know more about any of them, you'll find that their binomials are the key to almost everything that has been written about them. Having said that, I must admit that I can occasionally remember the common name of an organism when I can't pull the scientific name out of my memory banks. This is increasingly true with advancing years. As of November 2016, I am almost 83. Google and Wikipedia are wonderful memory aids which I, and I am sure all of you, use often. Many identification manuals list common names, and, therefore, they can in times of forgetfulness be one way to get at the scientific name. But that's as far as I am willing to go, unless you have to give a talk to non-scientists or small children, when common names are all they will be able or willing to accept. But why Latin or pseudo-Latin binomials? I can hear you saying plaintively. That's easy too. 1. Latin is officially a dead language, and although scientists do coin new words, the grammar, vocabulary, and usage will change much more slowly than those of all living languages. In a changing world, we need the relative stability of Latin for our scientific names. 2. The use of Latin for names and, until very recently, diagnoses of all new taxa also means that no one can be offended by being forced to use someone else's language. Latin has again become a useful international standard. I do have one problem with the Latin terminology, and it is that the same generic names can be used in different kingdoms. For example, there is a hyphomycete genus called Graphium, an asexual phase of an Ascomycete. See picture, below, left, and chapter. 4. There is also a genus of butterflies called Graphium, below, right. I do not think that this is necessary or reasonable, and I personally would disallow it, especially since the affiliations of kingdoms may change. The fungi are now considered quite closely allied with the animals, rather than the plants. Fig 1.22 Different examples of Graphium. Tail J. Graphium agamemnon. When this butterfly feeds, it flaps its wings so fast that it looks like a humming. It is unlikely that anything will be done about this in the near future, but you should be aware of the problem. Kingdoms, classification, nomenclature, and biodiversity. Biodiversity. Different strokes for different folks. For some people, place names are evocative, calling up vivid memories of past experiences. For me, it is the names of organisms I have seen that bring back those experiences. Metrosideros excelsa conjures up Christmas in Auckland, New Zealand, where the red flowers of this tree adorn beaches in December. Proteocanaroides takes me back to the amazingly rich fynbos plant community in South Africa, where this flamboyant shrub flourishes, and has been adopted as the national flower. Amphiprion places me on the barrier reef in Australia, where these agile little damselfish with blue-white stripes live unscathed among the tentacles of large sea anemones. Such organisms and the communities and ecosystems of which they are a part are the real reasons I leave home. I hope that some of you will also come to think of the biodiversity you encounter as a measure of your quality of life. Biodiversity was one of the buzzwords of the 90s and has continued to be a newsmaker in the new millennium. Biologists know from the earliest years of their training that the Earth is blessed with an amazing number of different living things. As you have just read, systematists have tried to catalog and describe all these riches, but with widely varying degrees of success. We know practically all the birds, about 8,000 species, and almost all the mammals, that inhabit the Earth. But we know only a small fraction of the arthropods and fungi. How can I make those two statements so confidently? Because although there are many ornithologists and mammalogists scouring the globe for new taxa, they rarely find any. At the other end of the scale, entomologists and mycologists find new taxa every day. They have so far described approximately 1 million insects and about 100,000 fungi, but it is obvious to the professionals who work in these areas that huge numbers of both groups have not yet been described, and new sequencing techniques are revealing thousands of new taxa in many habitats. I began my mycological career examining the sequence of fungi involved in the slow decomposition of Scots pine, Pinus sylvestris, needles. Almost immediately, I found several microscopic fungi which turned out to be new to science, and I had the privilege of describing these fungi. You can see some of them in Chapter 11 of www.mycolog.com. Since I moved to Vancouver Island, off the west coast of Canada, in 1994, I have seen many mushrooms that could not be identified using the existing literature. I am convinced that many of these fungi are actually undescribed. How many fungi are waiting to be discovered? David Hawksworth came up with an ingenious answer a few years ago. Noting that Britain is among the most intensively investigated areas on Earth for plants and for fungi, he pointed out that almost all the flowering plants in Britain are known and that there are about 2,000 species. Although the fungi of Britain are definitely not as completely known, as new ones are still being described, about 12,000 species have been recorded there. This gives a ratio of about 6 fungi to each plant species. Extrapolating, perhaps rather ambitiously, from Britain to the entire globe, Hawksworth suggested that because there appears to be about 250,000 species of flowering plants in the world, there are probably 6 times as many fungi, about 1,500,000 fungi, in fact. Even if this figure is an overestimate, and there are only 500,000 fungi, we have still described only 20% of the total, and a huge task lies before us. Chapter 1. If we accept this figure as a working approximation, and no one has yet come up with a different formula, it brings us to a realization that about 2 centuries of mycology have so far succeeded in describing only about 6%-7% of the world's mycota, a pretty shocking state of affairs. Over the years, I have been involved in describing probably 300 fungal taxa and have now basically run out of steam in this area of mycology. But even if all the mycologists alive today were to publish 100 species apiece, they would still manage to describe only about 300,000 taxa. Thus, it is a multigenerational task. The preceding discussion assumes a steady state in which no species are added, and none are subtracted, from the global total. However, we know that this is not the case. New species are constantly arising, albeit at an unknown rate, as a result of the combined effects of selection pressure and genetic recombination. Our own species, by sticking its fingers into every existing niche and ecosystem, as well as creating new ones, is undoubtedly providing the fungi with new challenges at every turn, and they are surely responding to those challenges by spawning new taxa. Now we come to the tragic bit. Human activities are undoubtedly driving some fungi into extinction. We don't know which or how many are being lost, and it is absurd for anyone to suggest that we are losing 2 species each week or 20 each day. Information on extinctions is extremely hard to obtain. How can you tell when a particular microscopic fungus, which can be detected only by culturing the soil, or a macroscopic fungus that may fruit only once in 20 years, has finally succumbed? Nevertheless, we have good reason to suspect that these things are happening. For example, the huge fruit bodies of Brachioporus, Oxyporus, Nobelissimus, a long-lived bracket fungus that grows only in old-growth forests on the west coast of North America. See Chapter 5, are seen less and less frequently, and, along with the forests in which it lives, this species may certainly be considered endangered. One of our problems in North America is that we do not have extensive records of the mycota from the past, a baseline with which present-day comparisons can be made. Fortunately, some European countries, with centuries of data collection to draw upon, have been able to document the decline in numbers of many fungi and have published what they call red lists. These lists highlight the increasing rarity of many fungi and the apparent disappearance of some. What does a red-bellied toad from North Vietnam have to do with preserving fungal diversity? Read on. Dr. Bob Murphy, then director of the Royal Ontario Museum's Centre for Biodiversity and Conservation Biology, reported in the Globe and Mail for July 12, 1999, that he had just returned from collecting herpetals, amphibians and reptiles, in the North Vietnam rainforest. His comments about his animals are worth repeating here, because they're beautiful, aesthetically appealing, wonderful animals. You can get them protected. If you can get them protected, you protect the forest. If you protect the forest, you protect everything that's there, especially the fungi myotalics, which are producing the majority of new pharmaceuticals that are coming out. You will find an echo of that statement in Chapter 24, in the discussion of antibiotics, immunosuppressants, and other fungal metabolites. Therefore, our efforts to collect and describe the world's mycota need to be redoubled. As you will learn from many of the other chapters in this book, and perhaps most accessibly from Chapter 24, many fungi confer enormous benefits to mankind. I am Kingdoms, Classification, Nomenclature, and Biodiversity. Not referring just to the producers of antibiotics such as penicillin and immunosuppressants, such as cyclosporin, but also to the myriad species which recycle organic matter, especially plant debris, and to the others that establish obligate mutualistic symbioses with many of our most important plants. This awareness of our ignorance has led to proposals to compile what are called Altaxa, Biodiversity Inventories, or ATBI for short. A meeting to discuss such an ATBI for a forested area in Costa Rica came up with a figure of $20 million for the fungi alone. Although the Costa Rican venture didn't fly, it spawned a marginally less ambitious project for the Great Smoky Mountains National Park in Tennessee. Setting other organisms aside, it has been estimated that there are 20,000 fungi in the park, of which only 2,250 have so far been described. Hey, that's more than 10%, so we are already ahead of the game. A 2-year pilot project aimed at refining sampling methods and data protocols was begun in March 1999, and mycologists everywhere will be watching with interest, or being co-opted, as this unfolds. To learn more about this project, visit the website http://www.discoverlife.org at blue_brochure.html. Less than 10% of the park's estimated 100,000 species are currently known. Known species 9,800, estimated species and GT 100,000 Fig 1.3. Vertebrates estimated total in park, 475. Number currently known, 450. Estimated total in park, 20,000 fungi. Number currently known, 2,250. Estimated total in park, 5,400. Plants. Number currently known, 2,816. Estimated total in park, 76,000. Invertebrates. Includes protists, number currently known, 4,280. Chapter 1. Further reading on classification and biodiversity. Ainsworth, G.C. 1976. Introduction to the History of Mycology. Cambridge, UK, Cambridge University Press. Carlisle, M.J., S.C. Watkinson, and G.W. Goudet. 2001. The Fungi. 2nd ed. New York, Academic Press. Cavalier-Smith, T. 2001. What are fungi? In the Mycota, edited by D.J. McLaughlin, E.G. McLaughlin, and P. A. Lemke, 3-37, Volume 7a. Berlin, Springer. Krausen, R.A. 1970. Classification and Biology. London, Heinemann. Dawkins, R. 1995. River Out of Eden, A Darwinian View of Life. New York, HarperCollins Paperback. ed. 1996. W. W. Norton. Hawksworth, D. L. 1991. The Fungal Dimension of Biodiversity, Magnitude, Significance, and Conservation. Mycological Research 95:641-55. 2004. Fungal Diversity and its Implications for Genetic Resource Collection Studies. In Mycology 50:9-18. Heckman, D.S., D. M. Gazer, B. R. Idell, R. L. Stauffer, N. L. Cardoz, and S. B. Hedges. 2001. Molecular Evidence for the Early Colonization of Land by Fungi and Plants. Science 293:1129-33. Hibbett, David S., Manfred Binder, Joseph F. Bischoff, Meredith Blackwell, Paul F. Cannon, Ove. E. Erickson, Sabina Hoondorf, ETAL 2007. A higher-level phylogenetic classification of the fungi. Mycological Research 111:509-47. James, T.Y., P. M. Letcher, J. E. Longcore, S. E. Mosley Standridge, D. Porter, M. J. Powell, G. W. Griffith, and R. Vilgalas. 2006. A molecular phylogeny of the flagellated fungi, Chitradiomycota. And description of a new phylum, Blastocladio Mycota. Mycology 98:860-71 doi, 10.3852. Jakes, H. E. 1946. Living Things, How to Know Them. Dubuque, Iowa, W. M. C. Brown. Jones, M. D. M., Iforn, C. Gadel Ha, M. J. Egan, D. Bass, R. Massina, and T. A. Richards. 2011. Discovery of novel intermediate forms redefines the fungal tree of life. Nature 474:200203. doi 10.1038/Nature 09984 SV. Phylum Cryptomycota. Kendrick, B. 1981. The History of Conidioid Fungi in Biology of Conidioid Fungi, edited by G. T. Cole and B. Kendrick, 3-18, Volume 1. New York, Academic Press. Kendrick, B. and T. R. Nagraj. 1979. Morphological Terminology in Fungi Imperfecti in the Whole Fungus, edited by B. Kendrick, 43-62, Volume 1. Ottawa, Canada, Ottawa National Museum of Natural Sciences. Kirk, P.M., P.F. Cannon, J.C. David, and J.A. Stolpers, eds 2001. Ainsworth and Bisbee's Dictionary of the Fungi, 9th ed. Egham, UK, CABI Bioscience, UK Centre. Lucking, R. S. Hoendorf, D. H. Pfister, E. R. Plata, and H. T. Lums. 2009. Fungi Evolved. Right on Track Mycology 101:810-22. Margulis, L. and R. Guerrero. 1991. Kingdoms in Turmoil. New Scientist, March 23, 46-50. Margulis, L. and K. V. Schwartz. 1982. 5 Kingdoms. San Francisco, Freeman. Miller, 0. K. Jr., T. Henkel, T. Y. James, and S. L. Miller. 2001. Pseudotolostoma, a remarkable new valvate genus in the Elaphomycetaceae from Guyana. Mycological Research 105:1268-72. Mueller, G. G. Bills, and M. Foster, eds 2004. Biodiversity of Fungi. Inventory and Monitoring Methods. Amsterdam, Elsevier/Academic Press. Kingdoms, Classification, Nomenclature, and Biodiversity. Orpin, C.G. 1975. Studies on the rumen flagellate Neocolimastix frontalis J. Gen Microbiology. 91:249-62. Patterson, D.J., and M. L. Sogan. 1992. Eukaryote Origins and Protista Diversity in the Origin. An Evolution of Prokaryotic and Eukaryotic Cells, edited by H. Hartman and K. Matsuno. 13-46. New Jersey, World Scientific Pub Co. Rogers, D.P. 1978. A Brief History of Mycology in North America. In Proceedings of the Second International Mycological Congress, edited by E. G. Simmons, 253-65. Tampa, University of South Florida. Ross, H. H. 1974. Biological Systematics. Reading, PA, Addison Wesley. Taylor, T. N., M. Krings, and H. Gerb. 2006. Hasilomonospora general ETSP November, a microfungus from the 400-million-year-old Reni Chert. Mycological Research 110:628-32. Whitaker, R. H. 1969. New Concepts of Kingdoms of Organisms Science 163:150-60. Wos, C.R., 0. Candler, and M. L. Wheelis. 1990. Towards a Natural System of Organisms. Proposal for the Domains Archaea, Bacteria, and Eukarya. Proceedings of the National Academy of Science, PNAS, 87:4576-79. 2. A Mixed Bag. Protozoan Pseudofungi Kingdom. Protozoa, the so-called slime molds. Phylum Mixostellata, Dictyostellata, Labyrinthulata. Plasmodiophorida. Pseudofungi Kingdom. Chromsta. Phyla Hyphocatriomycota and Mycota. True Fungi Kingdom. Eumycota. Phylum 1 Chitradiomycota, Phylum 2 Blastocladio Mycota. Phylum 3 Neocolimastygomycota. First, let me elaborate a little on the definition of fungi given earlier. Fungi, whether chromostom or eumycotan, are heterotrophic, non-photosynthesizing, eukaryotes that absorb their food, osmotrophic, typically at the many growing points of their rather diffuse, indefinite body, often called a thallus or mycelium, which is made up of fine branching tubes called hyphae. The wall of the tubes is mainly composed of chitin, eumycota, or cellulose, mycota, which offer protection from the outside world, and within this wall, the cytoplasm and nuclei live and move and are able to explore small areas of it. The humongous fungus see Chapter 11 manages fairly large areas, inside their apically extending, microscopic, hyphal tunnels and much larger areas by means of their detachable reproductive units, called spores. Before I discuss real fungi, 4 outsider phyla must be mentioned. These are the so-called slime molds, the inappropriately named Mixomycota, Acrasiomycota, Labyrinth and Plasmodium. They're slimy, but they're not moldy. Although they used to be thought of as fungi, and still appear in some current mycological literature, this is at least phylogenetically incorrect. Here's why. The assimilative or somatic phases of 3 of these 4 groups are basically amoeboid, where none of the 4 ever produces hyphae, a diagnostic feature of most true fungi, and the assimilative plasmodia have no cell walls. The names currently applied to some of these groups are misleading, in that they imply a fungal nature, so in 3 cases, I have supplied new names reflecting their non-fungal affiliations. I hope you will agree with me after you have read the thumbnail sketches below and compared them with my later descriptions of the wall-possessing hyphal fungi. These phyla are included in some mycology courses because some of them, particularly Mixostellata, tend to a mixed bag. Turn up when we look for fungi. If mycologists, who have historically looked after them, abandon them, what other group of organismic biologists will agree to add these organisms to their already crowded course schedules? Phylum Mixostellata, formerly Mixomycota or Mycetozoa. This is the only one of these 4 non-fungal phyla you are likely to find if you go out looking for fungi in autumn. The macroscopic, slimy, Amoeboid Plasmodium, the somatic or assimilative phase of the organism, is populated by diploid nuclei and oozes around in the soil or in decaying wood or other organic matter, eating, yes, actually engulfing or ingesting, bacteria and other tiny food particles. You can keep the plasmodia of some mixostellids as pets, crawling around in petri dishes and eating old-fashioned rolled oats. Eventually, the slowly pulsating plasmodium will dramatically metamorphose into a cluster of stalked, dry sporangia full of powdery resting spores. Spore formation involves reduction division, meiosis, so when each haploid spore germinates to release a cell that may be either amoeboid or biflagellate, this cell can act as a gamete. Fusion of 2 compatible gametes produces a diploid zygote which can then fuse or aggregate with other zygotes, begin to feed, and eventually grow into a plasmodium. Once more, a typical mixostellid life cycle is shown in Fig 2.1. Germination. Swarm cell. Older plasmodium beginning of fruiting karyogamy, zygote. Fig 2.1 Mixostellata, i.e. Fes cycle of Stemonitis fusca. Chapter 2. Phylum Dictyostellata, formerly Acrasiomycota. In the assimilative phase, Dictyostelium discoideum consists of independent, individual amoebae which feed by ingesting bacteria. The reproductive phase begins when the amoebae begin to secrete cyclic adenosine monophosphate, CAMP, a type of pheromone which causes the amoebae to be mutually attracted. They stream together and form aggregations called pseudoplasmodia, or slugs, and these differ from true plasmodia in that each amoeba retains its cell membrane. Each slug crawls around until dry conditions prompt it to undergo differentiation, heaping itself up and eventually forming a structure called a saurocarp, which has a slim cellulosic stalk and an expanded head containing spores. Dictyostelium has been used as an experimental organism by many scientists because it provides a simple system for studying differentiation. Fig 2.2. Slug. Pseudoplasmodium. Emerging amoeba. And LTIJE. ETAO_L/L. 0. IO. I aggregated OJ at 0 EMO-EB spore nucleus. Fig 2.2 Dictyosteliata, life cycle of Dictyostelium discoideum. A mixed bag. Phylum Labyrinthulata, formerly Labyrinthula mycota. The colorless colonies of Labyrinthula cause a wasting disease of eelgrass, Zostera, one of the few marine flowering plants. The spindle-shaped, naked cells of the colony live and move entirely within a network of narrow, tubular, polysaccharide sheaths which they themselves secrete. They release biflagellate gametes, and the zygote divides mitotically to generate a new colony, whose cells are presumably diploid. Most other members of this group are also marine parasitizing algae. Fig 2.3. Tubular sheath. Fig 2.3 Labyrinthulata, Labyrinthula. Phylum Plasmodiophorida, formerly Plasmodiophoromycota. All members of this group are obligate parasites. Plasmodiophora brassicae produces uninucleate, biflagellate primary zoospores which penetrate a root hair of its host, cabbage, Brassica oleracea. Inside, they grow into multinucleate but still microscopic primary plasmodia. These eventually develop a wall and divide internally into uninucleate secondary sporangia. These germinate, releasing 4 secondary, biflagellate zoospores which leave the host. These may also act as gametes, fusing in pairs, but soon infect a root again, developing within host cells into multinucleate secondary plasmodia. At maturity, these can cleave into uninucleate cysts, each containing a single spore, which can persist in the soil for many years. This parasite stimulates the cabbage roots to become grossly swollen, a serious disease condition known as club root. Fig 2.4. Chapter 2. Secondary - 8 and sporangia, I secondary - comma. Zoospores T \, comma, plasmogamy cysts in host cell karyogamy -- Plasmodium / Plasmodium eukaryote stage / After meiosis, nuclei invisible, __, comma / nuclei visible, __, meiosis. In the root / air. Fig 2.4 Plasmodiophorida, life cycle of Plasmodiophora brassicae. Kingdom Chromsta. Phylum 1, Hyphocatriornicota. This group is like the Chitradiomycota in many ways. They live in fresh water or soil, they can be parasites or saprobes, they may be holocarpic or eukarpic, the latter kind having assimilative rhizoids, and the sporangia release uniflagellate zoospores. So why aren't they Chitradiomycetes? A single characteristic visible under the light microscope gave the original clue and was soon supplemented by several ultrastructural features accessible only to the electron microscope. Fig 2.5. The visible feature concerns the behavior of the flagellum on the zoospore. In Chitradiomycetes, this propels the spore from the rear, but in Hyphocatriomycetes, the flagellum is seen to be attached at the front of the cell, where it works like a swimmer's arm or an oar. Other / comma / A mixed bag. Differences are revealed by the transmission electron microscope. In Chitradiomycota, the axis of the flagellum is smooth, a whiplash flagellum, while in the Hyphocatriomycota, the axis of the flagellum bears many fine lateral filaments called mastigonemes or flimers that give it the name tinsel flagellum. These may appear to be insignificant differences, but biologists consider the numbers and kinds of flagella on zoospores to be an extremely conservative characteristic, one likely to remain unchanged over vast stretches of time. perhaps even hundreds of millions of years. This makes it an important indicator, which is amply confirmed by the ultrastructural differences illustrated above, and justifies the recognition of phylum Hyphochitriomycota. There are few well-documented species in this phylum, but Hyphochitriomycotenoids is common in F underscore dot dot dash backslash ER dash N dot R dash slash 90 K 0 backslash LI degree Y backslash 1I backslash dot M and GTF idlim dot comma backslash ye dot T slash LLLN backslash single quote backslash LTRT1 RI slash G N slash T IM O R fill comma backslash RU backslash slash R and J pound NFC 11 and LT backslash comma 0 dash FF I backslash Q GJ slash colon slash LYL slash and LT slash Y L J colon TRS dash L slash F slash comma M backslash comma pound 5 TFLE and LTR backslash M RFJL underscore 0 T CL backslash dot RT backslash comma I slash dash 1 backslash dot comma slash dot dash comma colon LIV T backslash LCA D1 slash D backslash degree and LT backslash HR MJ ICEC co 03 dot FJ slash 1 F colon dot dash IGF slash T dash colon dot comma colon I C 11 Kiv dollar colon backslash FNFC if I colon I Hyphochitriales Spizellomyces Spizellomystales Rhizophidae Chitradiales TIH slash 2 1 hashtag dash H I backslash TD1 comma dash P dash comma underscore dash dot L dot RRU dash dot YT dash fluid dash L dash slash underscore comma colon underscore dot underscore Fig 2.5 ultrastructure of fungal zoospores ER endoplasmic reticulum F flagellum G Golgi apparatus K chyntusum L lipid M mitochondrion M microbody MN mastagonium MT microtubules N nucleus NC nuclear cap ribosomes NFC non-functional centrioles nu nucleolus R ribosomes Rho rootlet RU rumpusome SD striated disc L colon dot dash TI 1 dot backslash OJC E J1 and GT4 1F slash V slash I slash R backslash TN 1 backslash dash G dot CJ Z colon I TL I 2 and LT dash slash FJLCC equals colon dot dash colon R slash 1 dash and GT dash 1M V slash dot 1 colon T SDAJ 5E H I catenaria Blastocladiales Monoblepherus/A, Monoblepharidales. -er. _, /m, er, and lt, mv, g. and gt /l dash, /r, li /r, n, mt and gt /yt, 1r, /1, dash, comma, 11, and gti. 22 Chapter 2. Soil and is often, like the chytrid Spizellomyces and Chitradium, found in pollen when this is added to soil as bait in the laboratory. This species may be of some importance in the natural biological control of plant pathogenic Mycota, see below, by parasitizing their oospores. No hyphochytrid has yet been seen to sexually reproduce. Released. 7. Zoospores host. And Lt plus I. Branched aerial sporangiophores. / Aquatic zoosporangium. Sporangia. A. B. Detachable. / Host epidermis. T sporang 1A dash, 21. AC. / I / 1 0 W. 1, N. FOI. VR dash. OJ. Smoge. Branched. And LT. 1 VPL. NII_IL. Aerial sporangiophore. JJ repeatedly. Percurrent sporangiophores. Host tissue and fungal hyphae. 2. C. I. Fig 2.6 Asexual reproduction in Mycota. A. Saprolignales, BD, Paranasparalis. / A mixed bag. Kingdom Chromsta. PHYIUM2, Mycota. Having just read about the minute but basic differences between the two previous groups, you won't be surprised to learn that similar inconspicuous features distinguish the Mycota, our last and most important group of chromston fungi. Basic features: 1. Oomycetus zoospores have 2 flagella, 1 tinsel, 1 whiplash. Fig 2.6a, and these arise from the side of the cell rather than at the front or the rear, as in other groups. The zoospores swim with their tinsel flagellum pointing forward, while the whiplash flagellum is directed backward. 2. Unlike the nuclei of all true fungi, those in the assimilative hyphae of Mycetes are diploid. 3. The walls of the hyphae usually contain a cellulose-like material, a poorly crystallized hexose polymer, although this makes up only a fraction of the cell wall, and chitin has also been found in some Mycetes. All of these characteristics separate them from the Eumycotan fungi. But the feature that gives the group its name is its oogamous sexual reproduction and the fact that each zygote develops into a thick-walled, persistent oospore, Fig 2.7a, b. Note, however, that fungal oogamy is not unique to the Oomycota. Some Chitradiomycota such as Monoblepherus do it, too. Young. Oogonia antheridium b oospore. Fig 2.7 Sexual reproduction in Mycota. A. Saprolignales, b. Paranasparalis. Chitradiomycota and Hyphochitriomycota are often extremely inconspicuous, as befits organisms that in some cases devote an, albeit brief, lifetime to exploiting a single pollen grain. Mycota, although they also include some holocarpic and eukarpic unicels, often produce extensive hyphal networks, mycelia. These fungi, covering a dead fish like a whitish fur coat, as I saw on the dead salmon in the Adams River in 2010, or devastating crops such as grapes, hops, lettuce, cabbage, radishes, potatoes, and tobacco, have spawned a number of common names: water molds and white rusts, downy mildews, and damping off. Chapter 2. Germination. Asexual. Oogonium. Oosphere. Sexual. J. Oospores. Penetration of /Q fertilization 0 tube. 0 0 0. J. I. A. Caseagomerl. IIR. G. Plasmogamy. SIR. Fig 2.8 Saprolignales, life cycle of Saprolegnia. Order Saprolignales. The water mold, Saprolegnia parasitica, attacks fish and their eggs. After establishing itself, the fungus reproduces asexually. The tips of the normally non-septate hyphae become modified into long mitosporangia, Fig 2.8, delimited by a cross wall at the base. These asexual sporangia release biflagellate zoospores, often called swarm spores, which actively swim for a while and then encyst. A mixed bag. This means that they stop swimming and develop a thick wall. Later, they germinate again as secondary zoospores which, if they are lucky, will find a new substrate and develop into new assimilative thalli. This process of encystment, followed by a repetition of germination, is a strategy that gives the spores a second chance at finding food if they aren't so lucky when they are first released. Many Saprolegnia and Aclea species form compatible Antheridia and Oogonia on the same mycelium, which means that they are homothalic. Since the assimilative thallus is diploid, meiosis must take place inside the gametangia. Each globose oogonium contains several eggs, Fig 2.7a. A number of Antheridia may grow toward and touch a single oogonium, penetrating its wall at preformed thin spots and sending in fertilization tubes which deliver the male nuclei to the eggs. Fertilization is more reliable because neither gamete is exposed to the vagaries of a free-swimming existence. The whole double life cycle is illustrated in Fig 2.8. The zygotes develop thick, resistant walls and obviously function as survival spores that can live through such catastrophes as the drying up of a pond or stream. Homothalic species have lost the enhanced variation provided by outbreeding, but they still benefit from the thick-walled resting oospores produced by the sexual cycle. Order Paranasparalis. Many members of this order are obligately parasitic on higher plants. In some cases, they cause epidemics that devastate important crops. The buildup of these epidemics is made possible, 1. By our need to grow dense stands of single plant species, monoculture crops, and, 2. By aerial transmission of the fungi, which have evolved airborne mitosporangia, Fig 2.6b, c. Note that these are often wrongly called conidia, as they are analogous but not homologous to those spores, which are discussed under the subkingdom Dicaria. Oogonia are also formed, each containing a single egg, Fig 2.7b. Sexual reproduction is usually homothalic, the Antheridium arises from the same thallus as the Oogonium. We will examine representatives of 5 genera from 3 families, Pythiaceae, Pythium, Phytophthora, Paranasparaceae, Paranaspara, Plasmopara, and Albugenaceae. Albugo. Damping off disease of seedlings, Pythium Pythiaceae. This is a soil-borne disease, so its causal agents, species of Pythium, have no need for airborne sporangia, since they persist saprobically in most soils and spread by zoospores during wet conditions. When these motile cells find young plants, they cause infections which release toxins and also produce a pectinase enzyme which dissolves the middle lamella, which glues plant cells together. Seedlings of many plants collapse rapidly when this disease strikes at the base of their delicate shoots. Damping off is, unhappily, familiar to gardeners who try to get a head start on the growing season by germinating seeds indoors. The disease can be controlled by using heat-sterilized soil, by dusting seeds with Benamil, a very safe fungicide, although now delisted, see Chapter 13, or by watering seedlings with other fungicides such as Zenab or no damp off. 26 Chapter 2. Vesicle. Encysted zoospores. Sporangium. Me. Somatic. Nation. Hypha. Germination. Antheridium. 0. 6GF. JA. Oospore A.I5CTJ. Karyogamy plasmogamy. Fig 2.9 Paranasparalis, life cycle of Pythium. A mixed bag. Late blight of potato, Phytophthora. In the mid-19th century, a North American umi seed caused havoc in Europe. Unintentionally imported to Ireland and reintroduced to a susceptible host from which it had been separated 250 years earlier, Phytophthora infestans wiped out the Irish potato crop in the damp, cool summers oft years 1845 to 1847, causing widespread famine in that poor, one-crop economy. At first, the disease was not attributed to the fungus, but to an excess of water in the plants, or even to the effects of newly introduced steam locomotives. However, the Reverend M.J. Berkeley, England's leading mycologist, drew sporangiophores emerging from the potato leaves and correctly concluded that the fungus caused the disease. His contemporaries eventually admitted that he was right, and this episode led to the founding of the discipline now known as plant pathology. The ravages of potato blight contributed to 1 million deaths and drove millions more to emigrate from Ireland. 10 years after the first epidemic, the population of Ireland had crashed from 8 million to 4 million. Contemporary pictures from the London Illustrated News convey some of the misery caused by the recurring epidemics. Ragged and starving peasant girls gleaned desperately in the fields for anything edible. And whole families were forced to leave their homes forever. Some died. Many sailed for North America, but 187 doubly unfortunate souls were shipwrecked and drowned off Farallon National Park near the tip of the Gaspé Peninsula, Quebec inside of the Promised Land. There are websites that explore the potato famine, easily found on Google. Until 1976, this heterothallic pathogen, except in its homeland, Mexico, was asexual, representing only one of the two mating types. In that dry year, many crop failures led to importation of potatoes that carried the other mating type with them. It has also been suggested that new genotypes were spread in seed tubers, in tomato tissues, and even in tropical storm systems. However it came about, new sexual populations have certainly supplanted the older asexual strain and have led to a resurgence of the disease, which now costs the United States alone about $3 billion per year in attempts at control by spraying fungicides and by trying to breed resistant varieties of potato. The airborne sporangium of the potato blight fungus is clearly an efficient short-range dispersal mechanism, although humans were just as clearly the long-range vectors of this disease. Potato blight is still a threat, although it can be controlled by 1. Spraying with fungicide at times carefully chosen by plant pathologists, nowadays with the aid of special blight forecasting computer programs, 2. Destroying infected foliage before harvest, and 3. Planting disease-resistant seed potatoes. In addition to its most infamous species, the genus Phytophthora contains 60 other species. Some of them are also, as one might suspect, serious plant pathogens. Phytophthora soji plagues soybean farmers in North America. Phytophthora megakarya attacks cacao trees in West Africa. A hybrid between P. cambivora and P. fragarii has killed about 10% of the alder trees, Alnus species, in Britain and is now spreading to France, the Netherlands, Sweden, Germany, and Austria. Phytophthora cinnamomi is destroying the jarrah, eucalyptus, forest and other natural ecosystems in Western. Chapter 2. Australia and cork oak forests in Spain, and it is a serious problem for growers of avocados, pineapples, and ericaceous shrubs elsewhere in the world. No wonder this genus provides work for hundreds of plant pathologists and mycologists worldwide. Downy mildew diseases, P/Asmapara, Paranasparalis. The downy mildews include blue mold of tobacco and downy mildew of grape. Since these diseases have historical or economic importance, I'll tell you something about them. In this group, the mitosporangia are no longer unspecialized hyphal tips, but are borne on highly differentiated, branched, aerial sporangiophores. The sporangia don't only release zoospores but are themselves set free and blown or splashed away. The sporangia of Paranaspara germinate by producing a hypha, although those of most other members of the group still release motile zoospores. Downy mildew of grape, P/Asmapara Paranasparaceae. Plasmopara viticola, an umi seed native to America, causes downy mildew of grapes. It can be found attacking wild grapes every summer. But because it evolved along with its North American host, a biological balance has been struck, and the wild Vitis species aren't seriously damaged. When this fungus was accidentally introduced to Europe in the 1870s, it was a different story. The French grapevines, Vitis vinifera, had no resistance to the new pathogen and were quickly devastated. Fortunately for the enophiles, wine lovers, oft-world, the concoction of Bordeaux mixture, one of the world's first practical fungicides, by a university professor, yes, we profs occasionally have good ideas, saved the day. The rather strange story behind this invention is told in Chapter 13. The life cycle of Plasmopara is illustrated in Fig 2.10. Look at this set of diagrams carefully and decide at which stage you think it would be most vulnerable to chemical attack. Here's a clue, the answer begins with Z. Blue mold of tobacco, Paranaspara Paranasparaceae. My last example of a downy mildew fungus is Paranaspara tobacina, which causes blue mold of tobacco. This disease was first recorded in Ontario in 1938, was epidemic in 1945 to 1947, and had not been seen since 1966. But in 1979, seedlings infected with the pathogen were imported to Canada from the United States, escaping detection at the border. The weather that year favored the development and spread of the fungus, and a large-scale epidemic ensued. About 30% of Ontario crop, worth $100 million, was lost. Blue mold helped to put the Ontario tobacco industry on a slippery slope, and the decline in tobacco acreage is still continuing, although it is now driven by changing societal attitudes towards smoking. Paranaspara once again became a serious problem, a coffin nail, for Ontario tobacco growers in 1997. White rust disease of crucifers, A/Bugo Albugenaceae. This disease attacks all members of the family Brassicaceae such as cabbage and radish and is caused by Albugo candida, which produces extensive white blisters on leaves and stems. See www.mycolog.com. These unique blisters contain innumerable unicellular. A mixed bag. Sporangiophore with sporangia. Zoospore. Sporangial germination. / Oospore germination. Zygote. Oospore. Fig 2.10 Paranasparalis, life cycle of Plasmopara viticola. Mitosporangia developing in chains from the tips of short, tightly packed sporangiophores. Fig 2.6d. When the host epidermis bursts, the sporangia are dispersed by wind or rain splashes to other host plants, where each can germinate to release 8 biflagellate zoospores. Oogonia develop later, inside the host stem or leaves, and sexual. Reproduction is usually heterothallic, or outbreeding. Chapter 2. True Fungi Kingdom Mycota. As I mentioned in Chapter 1, what we call fungi share many morphological and behavioral similarities in their assimilative phase, but they do not have a uniform genetic background. It now seems obvious that they have evolved from at least 2 ancestral lineages. Some fungi can produce cells that swim by means of 1 or 2 very fine whip-like extensions called flagella, homologous to the tails of sperms. 5 phyla Hyphochitradiomycota, Mycota, Chitradiomycota, Blastocladio and Mycota fall into this category. But molecular evidence tells us that they are members of 2 different kingdoms, the first 2 being chromostom in origin, and related to the brown algae, and the last 3, despite their flagellate phase, Eumycotan. The other 6 generally recognized true fungal phyla never have motile cells, and they the Microsporidia, Zygomycota, Cryptomycota, Glomeromycota, Ascomycota, and Basidiomycota make up the rest of kingdom Eumycota, although only the Ascomycota and Basidiomycota produce highly differentiated, multicellular reproductive structures, as you will see. Before I introduce you to the three flagellate Eumycotan phyla, a few words about Flagella, singular, flagellum. These are very long, narrow organelles, essentially contractile extensions of a cell, which have the ability to beat or make whip-like motions that confer motility to the cell. Flagella move the cell about in water, giving it the ability to swim up chemical gradients, that is, to move from a lower concentration of a substance toward a higher concentration, such as those which lead toward a sexual partner or a suitable host organism. The amazing thing about flagella is that wherever we find them among eukaryotic organisms, they have essentially the same fine structure, consisting of 9 pairs of peripheral microtubules that run along the flagellum shaft and 2 central microtubules. The 9+2 pattern. Each microtubule is built from a protein called tubulin, which has subunits that are arranged in 13 vertical stacks, count them in Fig 2.11, around a hollow center. Fig 2.11 transverse section of a typical 9+2 microtubule flagellum, diameter about 170 nanometers. A mixed bag. This image of a transverse section of a flagellum, which is about 1/60.16 of a micron thick, 100,000 of them laid side by side would be less than an inch thick, would look more or less the same whether the flagellum came from one of the simple fungi discussed here or from a protozoan such as Paramoecium, which has lots of short flagella called cilia all over the outside of the cell, or from a unicellular or colonial green alga such as Chlamydomonas or Volvox, or a sea gooseberry, a member of phylum Ctenophora, which has plates of cilia that beat in rhythm and move the whole macroscopic organism, or from the sperm, male gamete, of a brown alga, a sea urchin, a moss, a fern, or a human. You see, all eukaryotes really are related. By the way, the flagella of bacteria are completely different, but that's another story. Kingdom Eumycota. Phylum 1, Chitradiomycota. The somatic phases of the microscopic Chitradiomycetes vary widely in appearance, but when they undergo asexual reproduction, most produce zoospores with a single, backwardly directed whiplash flagellum. I will discuss members of 3 orders, the Chitradiales, Spizelomystales, and Monoblepheridales. Although these orders were formerly separated by the morphology of their microscopic thalli, we now know that this is too variable to be reliable. Emphasis has switched to ultrastructural features of the zoospore, some of which are illustrated in Fig 2.5. Unfortunately, although conservative, and therefore taxonomically valuable, these characteristics can be seen only in the transmission electron microscope after elaborate preparative techniques. Fortunately, you can see enough of an Allomix zoospore under the light microscope, using phase contrast optics, to identify the phylum, although some members of this phylum recently discovered in the rumen of large herbivorous mammals have many flagella on each cell. Orders Chitradiales and Spizelomystales. These orders look very similar under the light microscope, and it takes an expert to tell them apart. However, most Chitradiales are aquatic, while most Spizelomystales live in soil. Until important differences were found in the ultrastructure of their zoospores, Fig 2.5, the 2 orders were considered to be one. These simple fungi do not produce hyphae. They are often parasitic, and their assimilative thallus often consists of a single cell. This cell is either 1. Entirely converted into a reproductive sporangium, the holocarpic mode, Fig 2.12b, as in Olpidium brassicae, or 2. Differentiated into assimilative rhizoids and a sporangium. The eukarpic mode, Fig 2.12a, as in Chitradium lagenaria or Spizelomyces punctatus. Other chitrids have a more extensive system of rhizoids, called a rhizomycelium, which may nourish several sporangia, as in Cladochitrium, Fig 2.12c. We describe this multisporangial condition as polycentric to differentiate it from the monocentric forms just mentioned, which produce only a single sporangium. The difference between rhizoids and rhizomycelium is that rhizoids generally have no nuclei in them and are usually less than a millimeter long, while rhizomycelia contain nuclei and can be much more extensive. Chapter 2. You might be interested in the activities of some of the members of these two orders, including the fungi just named. The Eukaryotic Spizellomyces punctatus and Chitradiomycosis parasitize pollen grains. The polycentric Cladochitrium is saprobic, growing on decaying aquatic vegetation. At least one chytrid, Syncytrium endobioticum, is a serious problem for farmers because it causes wart disease of potato. This fungus produces dark brown, cauliflower-like growths on the tubers and a catastrophic reduction in yield. Fortunately, although the pathogen is widespread in Europe and has spread to Newfoundland, resistant varieties of potato help to keep the disease under control. Other microscopic chytrids parasitize algae, see the website, and can be so numerous as to cause epidemics which significantly, if temporarily, reduce primary production in lakes. The frog problem. Most recently, a Eukaryotic chytrid has been found attacking amphibia in many regions of the world. It has been associated with significant die-offs of frogs in Australia, in Central America, and at the National Zoo in Washington, D.C. The condition has been quite reasonably called Chitradiomycosis since no other organisms are yet known to be involved. The fungus lives inside epidermal cells and causes thickening of the skin, which may interfere with normal respiration, as frogs breathe partly through their skins. Its zoospores have ultrastructural characteristics, the kind that can be seen only under a transmission electron microscope, that put it into a new genus, Batrachochytrium. Joyce Longcore, the chytrid taxonomist who determined this, published a short article in Inoculum, the newsletter of the Mycological Society of America, for October 1998 and gave illustrations of both the Eukaryotic thallus and the zoospores. The study describing the new genus appeared in Mycology in April 1999, and the full reference is given at the end of this chapter. No one knows why this fungus would suddenly begin killing frogs in places as diverse as Australia and Panama. The fungus may have been transported to these places only recently, perhaps even on the boots or equipment of researchers studying the disappearance of frogs. It may be the case that the fungus was present for a long time, but frogs are now succumbing because their immune systems have been impaired by recent environmental changes. One obvious change is increased ultraviolet, UV, light, which is known to damage the immune systems of animals. Recently, chlorinated chemicals released by humans have attacked the ozone layer in the upper atmosphere, allowing 10% more UV light to reach the Earth's surface. Industrial chemicals may also be damaging the frogs' immune systems. Retinoids are under suspicion because they cause birth defects in many animals, including frogs and humans. Accutane, used to treat acne, is a retinoid known to cause birth defects in humans. If you are interested in pursuing this topic, I suggest you search for more information on the internet. Check out the archives of Rachel, an internet environmental magazine. Sexual reproduction in chytrids and Spizellomycetes should be re-examined, as it used to be assumed that any zoospore with 2 flagella and every resting spore resulted from nuclear fusion. Now we know that some biflagellate zoospores originate by a mixed bag. Incomplete differentiation of the cytoplasm during zoospore formation, and that many resting spores are just thick-walled asexual sporangia which can survive dry periods. Order Monoblepherus. Monoblepherus Polymorpha, Figs 2.5, 2.12d, which is found on twigs of birch, ash, elm, or oak submerged in slightly alkaline freshwater pools, is the first fungus we have met that has gone all the way to complete sexual differentiation of gametes. The male gamete is motile, a sperm, but the female, an egg, is Eukaryotic thallus. Zoospores rhizoids A, Spizellomyces C, Cladochitrium D, Monoblepherus holocarpic thallus zoosporangia root cell amoeboid zygote B, O/Pydium and C stead zygote zoospores. Fig 2.12 Types of thalli and reproductive structures among the Chitradiomycota. A. Eukaryotic thallus of Spizellomyces punctatus, Spizellomyces, in pine pollen. B. Holocarpic thallus of 0/Pydium brassicae, Chitradiales, in a cell of cabbage root. C. Polycentric thallus of Cladochitrium, Chitradiales. D. Stages of oogamous reproduction in Monoblepherus Polymorpha, Monoblepherus. Chapter 2. Not. This style of sexuality is called oogamy. Sperms form in gametangia called antheridia, while eggs develop in oogonia, which are found on the same hypha just below the antheridia. Sperm is often released before the adjacent oogonium is ripe. This may be a mechanism for avoiding self-fertilization and thus ensuring outbreeding, called heterothallism. In fungi, after the egg has been fertilized, the resulting zygote becomes amoeboid, moves out onto the top of the oogonium, and encysts, developing a thick wall. Meiosis probably occurs when this resting spore germinates, producing a germ tube, another name for a first hypha. Although the Chitradiomycota vary in many things, such as in the morphology of their assimilative phase, in their patterns of sexuality, and in their adoption of parasitic or saprobic lifestyles, most of them have motile spores, zoospores or gametes, with one flagellum at the back, posteriorly uniflagellate. The cell swims like a sperm. In addition, their cell walls, like those of the other, more complex, eumycotan fungi, are largely made of chitin, a polysaccharide very similar to the stuff of which insect exoskeletons are made. They synthesize lysine by the same pathway, see Chapter 8, and the more advanced members of the group produce true hyphae. The Chitradiomycota apparently represent modern survival of the ancestral line that evolved into the eumycotan fungi. Perhaps surprisingly, two presumed members of the Chitradiomycota, the genera Olpidium and Rosella, have been discovered to be completely separate clades. Indeed, Rosella was found to be the earliest diverging lineage in the fungi and perhaps a member of yet another new phylum, the Cryptomycota. The parasitic Olpidium appears closely related to the Zygomycota. Kingdom Eumycota. Phylum 2, Blastocladio Mycota. This phylum was long considered no more than an order within the Chitradiomycota. But in 2006, the order Blastocladiales was promoted to the rank of phylum by James et al. From molecular evidence. Here, the thallus has both broad true hyphae and narrow rhizoids. Allomix Arbusculus, whose life cycle is illustrated in Fig 2.13, exhibits what we call a rotation between haploid and diploid thalli. Haploid thalli produce gametes in specialized gametangia, while diploid thalli produce flagellate zoospores and resting sporangia. In Allomix, the gametes come in two sizes, which is a condition called anisogamy. The general principle underlying anisogamy is division of labor, whereby the smaller, more mobile gamete, which we can now think of as male, actively seeks out the larger, less mobile, female, gamete, which has sacrificed some speed in order to carry enough food to give the next generation a good start. In Allomix Arbusculus, both kinds of gamete are formed on the same haploid thallus. The colorless female gametangia are born at the tips of hyphal branches, with the orange male gametangia immediately below. Zygotes develop into diploid thalli which bear two kinds of sporangia, thin-walled and thick-walled. The nuclei of thin-walled sporangia undergo repeated mitosis and a mixed bag produce metospores, which in this case are diploid, uniflagellate zoospores that can establish new diploid thalli. The other kind of reproductive structures, resistant sporangia, shown on the right side of the diagram, are thick-walled, brown, and can survive for up to 30 years. Eventually, some environmental stimulus triggers reduction division, meiosis, in these sporangia, and the resultant haploid myospores are liberated and develop into sexual thalli. Chylomomyces is another genus of the Blastocladio Mycota, in which some species are obligate parasites of mosquito larvae, and attempts are being made to use them in biological control of these insects. See Chapter 14. Chapter 2. Kingdom Mycota. Phylum 3, Mycota. Just to spoil our picture of the Chitradiomycota even further, in 1975, Orpin discovered some new and very different Chitradiomycetes fungi living in the rumens of large herbivorous mammals. These fungi, mostly species of Neocolymastics, were obligately anaerobic. They resembled their aerobic relatives in many ways but had no mitochondria and often had multiflagellate zoospores. 15 species of anaerobic chytrids had been described by 1994. They produce rhizomycelia which efficiently penetrate plant material and have enzymes that more effectively break down cellulose than the cellulases of the mold Trichoderma. See Chapter 14 Part 3 and Chapter 24. They are now classified as the order Colymastigales in phylum Neocolymastigomycota. Further reading. Barr, DJS 1990. Phylum Chitradiomycota in Handbook of Protoctista, edited by L. Margulis. J0. Corliss, M. Melkonian, and D. J. Chapman, 454-66. Boston, Jones and Bartlett. Barron, G. L. 1991. Protoplasm in Motion Seasons 31, No. 2:20-25. Bigelow, D. M., M. W. Olson, R. L. Gilbertson. 2005 Labyrinth Holaterastries sp. November, a new pathogen of turfgrass Mycology 97:185-90. Bonner, J.T. 1967. The Cellular Slime Molds. 2nd ed. Princeton, New Jersey, Princeton University Press. 1993 Life Cycles, Reflections of an Evolutionary Biologist. Princeton, New Jersey, Princeton University Press. Buzzacki, S.T., ed. 1983. Zoosporic Plant Pathogens, A Modern Perspective. New York, Academic Press. Cavender, J.C. 1990. Phylum Dictyostellata in Handbook of Protoctista, edited by L. Margulis. J0. Corliss, M. Melkonian, and D. J. Chapman, 88-101. Boston, Jones and Bartlett. Dick, M. W. 1990. Phylum Mycota in Handbook of Protoctista, edited by L. Margulis, J0. Corliss, M. Melkonian, and D. J. Chapman, 661-85. Boston, Jones and Bartlett. Dilusky, D.P. 1990. Phylum Plasmodiophoромycota in Handbook of Protoctista, edited by L. Margulis, J0. Corliss, M. Melkonian, and D. J. Chapman, 399-416. Boston, Jones and Bartlett. Eichinger, L., ETAL 2005. Isle Genome of the Social Amoeba Dictyostelium discoideum Nature 435:43-57. Irwin, D.C., and 0. K. Ribeiro. 1996 Phytophthora Diseases Worldwide. St. Paul, Minnesota, APS Press. Frederick, L. 1990. Phylum Plasmodial Slime Molds, Class Myxomycota in Handbook of Protoctista, edited by L. Margulis, J0. Corliss, M. Melkonian, and D. J. Chapman, 467-83. Boston, Jones and Bartlett. Fuller, M.S. 1990. Phylum Hyphocatriomycota in Handbook of Protoctista, edited by L. Margulis. J0. Corliss, M. Melkonian, and L. J. J. Chapman, 380-87. Boston, Jones and Bartlett. Fuller, M.S., A. Jaworski, eds. 1987. Zoosporic Fungi in Teaching and Research. Athens, Georgia. Southeastern Publishing Co. A Mixed Bag. Gray, W.D., and C.J. Alexopoulos. 1968 Biology of the Myxomycetes. New York, Ronald Hagiwara, H. 1989. The Taxonomic Study of Japanese Dictyostellid Slime Molds, Tokyo, National Science Museum. James, T.Y., P.M. Letcher, J.E. Longcore, S.E. Mosley, Standridge, D. Porter, M.J. Powell, G.W. Griffith, and R. Vilgalas. 2006 A Molecular Phylogeny of the Flagellated Fungi, Chitradiomycota. And Description of a New Phylum, Blastocladio Mycota, Mycology 98:860-71 doi, 10.3852. Kading, J.S. 1977. Chitradiomycetera Myconographia. Vedas, Liechtenstein, Kramer. Large, E.C. 1962. The Advance of the Fungi. New York, Dover. Longcore, J.E., A.P. Pessier, D.K. Nichols. 1999 Batrachochitrium dendrobatitis general ETSP November, a chytrid pathogenic to amphibians, Mycology 91:219-27. Margulis, L., J. 0. Corliss, M. Melkonian, D. J. Chapman, eds. 1990. Handbook of Protoctista. Boston, Jones and Bartlett. Moore, R.T. 2000. Mycological Dispatches about Phytophthora-Mycologist 14:93. Muhlstein, L.K., D. Porter, F.T. Short. 1991 Labyrinthula Zosteri sp. November, the causative agent of wasting disease of eelgrass, Zostera marina, Mycology 83:180-91. Olive, L.S. 1975. The Mycetozoans. New York, Academic Press. Orpin, C.G. 1975. Studies on the rumen flagellate Neocolimastix frontalis, Microbiology 91, No. 2, 249-62. Porter, D. 1990. Phylum Labyrinthula Mycota in Handbook of Protoctista, edited by L. Margulis. J. 0. Corliss, M. Melkonian, and D. J. Chapman, 388-98. Boston, Jones and Bartlett. Quammen, D. 2000. The Boilerplate Rhino, Nature in the Eye of the Beholder. New York, Scribner. Sparrow, F.K. 1960. Aquatic Phycomycetes. 2nd ed. Ann Arbor, University of Michigan Press. Spencer, D.M., ed. 1981 The Downy Mildews. New York, Academic Press. Webster, J. 1980. Introduction to Fungi. 2nd ed. Cambridge, UK, Cambridge University Press. 3. Eumycotan fungi, the mainstream and others. Phylum 4 Zygomycota, Phylum 5 Glomeromycota, Phylum 6 Microsporidia. First, a lean item display of the classification to show what goes where. Kingdom Eumycota. Phylum 4 Zygomycota. Subphylum Mucoromycotina, Order Mucorales. Order Endogonales, Order